Okay, so happy Wednesday, everybody. Hope you're doing well. So back to some Biology 107. We'll pick up where we left off here. We were talking about nomenclature, and this was the last slide that I showed everybody. So we were talking about the binomial nomenclature system. Uses two names. The names are usually Latin, but sometimes they're inspired by other things. And you're going to have your genus and species. So this is a big reminder that on all of your reports and all of your work in this course that you do this properly. So you're going to have two names, the genus name and the species name. Both of them are going to be in italics. If it's handwritten, you can underline it instead. That is totally acceptable. You can use abbreviations, so the genus can be abbreviated by letter. So also wanted to talk a little bit about virus nomenclature. So virus nomenclature is a little more loose. Sometimes you see it in italics and sometimes you don't. I think generally it's not in italics, and I'm not really sure why that is. Usually the way that viral nomenclature works is that there's some sort of descriptor. So the descriptor could be the name of the disease, or in some cases, it's the name of a place. You don't see that so much anymore, but anything that was named more than 20 years ago, it was actually relatively common to name a virus after the location it was first discovered. So if there was a new virus in Fort McMurray, it might be called the McMurray virus or something like that. Here's some examples. So these ones are named after the diseases. So rabies virus. Sometimes also there will be no space between the descriptive word and the word virus. So influenza virus can be one word. Sometimes there's different versions of the virus. So you can see influenza. There's actually A, B, and C. And so the ones that usually infect humans are A and B. A a little bit more common than B, and C not as common. Herpes simplex virus. So as you can imagine, that causes a type of herpes, so that would be oral herpes, which is also known as cold sores. Sometimes named after scientists, so Epstein-Barr virus, that's two scientists, and they both, I don't think they actually discovered it, but I think that they studied it and learned that it is the virus that causes infectious mononucleosis, so mono, if you've ever had that. Ebola, you may have heard of that one. It's named after a river in the Democratic Republic of the Congo. And Norwalk virus, this was named after, I think it's in Ohio, a town in Ohio. In fact, they don't like that name so much because who wants a virus named after them, right? Particularly one that's causing explosive diarrhea. And then, of course, the one on the right, this is the one that's been causing us all the trouble this year. And so the abbreviation for this virus is SARS-CoV-2. So SARS stands for the disease symptoms, so severe acute respiratory syndrome, and CoV stands for coronavirus. And the word corona, in fact, it means something like crown. And so if you look at some of the electron micrograph images of coronaviruses, they have the little spikes and that kind of look like jewels on a crown. So it was named by a woman in the 1960s who thought, hey, this looks like a crown. So the word coronavirus came out. So missing something here. Let me just feel like maybe I've loaded up the wrong slide. So just give me a moment here. I'm going to take a look and see what I am missing, what I thought I'm missing. So hold on a second. Oh, there it is. Okay, I see what's going on. Okay, yeah. We'll come back to the T-Rex in a minute. I'm just going to talk about a couple other things. I have a Kahoot coming up, and the T-Rex is going to show up in the Kahoot. But a couple other things I want to talk about first. So in the Kahoot, by the way, we will look at there'll be some name examples. So let me just share this with you. So part 2 of topic 1 is kind of just an introduction to science and the scientific method. And so hopefully some things you've seen before. There's going to be some overlap in material with what we talk about in the lab. And so what is science? You know, a lot of people will associate science with knowledge, but there's many types of knowledge out there. You can know things without using science. Maybe your mom loves you. How do you know that? It's not like you're doing experiments in a lab to figure that one out. There's knowledge that can be logic, mathematical knowledge, those kind of things. So science is based around observation of the natural world, and often is tied to experiments as well. So if you think about science, there's actually two types of primary types of science with lots of overlapping things going on in between. And so most people think about the experiment part and the hypothesis stuff, but a big part of science is actually what we call is discovery science. So in that case, and I'll have some examples in the next slide, but this is mostly just looking at things and observing things. So, you know, maybe you've discovered a new species of squirrel, right? And you can describe it. You can say it's this big, it's this color, this is what it eats, and so on, right? And the one that we talk about a lot is, of course, in school, we talk about hypothesis and experiment-driven type of science. So discovery science, like I said, we're describing things and maybe not necessarily doing experiments, right? So analyzing genomes, like I said, new species of squirrel, or maybe you've stained a specimen under a microscope and you've discovered a new organelle. And that's kind of what discovery science is. So it's a huge, really big, important part of science. It's and we can describe the natural world through it. A really powerful type of science, of course, is the hypothesis-driven science. And maybe you've seen flow charts that are kind of like this. You know, this whole idea where you ask a question and maybe form a hypothesis. And I'll talk about hypotheses in a moment. And then you might design an experiment that can test or confirm that hypothesis. And if it's supported, then, you know, you can maybe make some conclusions. If not, maybe you try a different experiment, all those kind of things. It's not necessarily that simple, right? And I like this little diagram here because it's actually showing that it's quite complex. And just a couple of things I want to point out, right? So if your hypothesis is not supported, right, by the data, so maybe my hypothesis is this new squirrel eats, let's say, acorns, right? And so I watch the squirrel and I don't see it eating any acorns. So then maybe I can make a new hypothesis. Maybe it eats pine cones or something like that, right? So that's kind of a big part of the scientific process. Another thing to mention is it's not linear necessarily, right? Very rarely do experiments work out as planned. And if you ever get a chance to work in a lab, you'll see that you're repeating things constantly because you're tweaking this and you're tweaking that, and it can be very, very complex. If you've ever done a science fair project, it's never simply a one-shot. It's always very, very complex. I think I have a couple other notes here. And then, of course, you know, if you think your hypothesis is supported, usually it leads to some sort of publication or consultation with other people, and they may disagree with it. Maybe they think your data is not good. Maybe they think your experiment is sloppy. So sometimes things don't necessarily end up in the scientific record just because they've been experimented on. Often it takes years to really get a lot of good data on things. And science is never done. It seems to go on and on and on. We can always learn new things. And, you know, hypotheses are revisable, and sometimes people come back and re-explore them later on. So just some things to think about with science. I have a couple of historical experiments to share with you where we're going to talk about this a little bit more. Let's talk about what a hypothesis is. If you look it up on you know, Google or something like that, or in your textbook, there's a lot of different ideas around it. But this is probably the best answer here, is that it's a hypothetical explanation for something, right? So my hypothesis is that the squirrels live on nuts. That's not too bad. What am I explaining? I'm explaining what they eat, right? The best hypotheses are testable or and also falsifiable. So that means if I have a hypothesis, a good one is something that we can actually test, right, by observing or doing an experiment. A bad hypothesis is something we can't test, right? And you know, just trying to think of an example of a bad hypothesis. there's lots of them going out there. For example, regarding the pandemic, right? You know, the hypothesis that you know, that the virus was made in a lab. You know, it's not exactly you know, something that's easy to test, and in fact, there's no data to support that. And if you want to talk about that further, I can get to that. So I'm going to actually give you guys a quick Kahoot right now. I think I got 4 or 5 questions. So load up the app. I'll answer some questions, and then I want to get back to what is science and some scientific examples. So load up Kahoot, and I am going to run it. There we go. I think you should be able to see that. It looks like we're good. Oh, okay, I guess I gotta load it up. Look for the game pin. I'll give you guys about a minute to join. 558-7440. Okay, here we go. See who can get the most points. What does a eukaryote have that a prokaryote does not? Okay, you got to be quick on some of these. That one you only had 10 seconds. So all the people answered, got it correct. The eukaryote has a nucleus. Remember, eu means true and karyote means nucleus. All right, good job, Michelle. Number 2, which of the following are proper ways to name this dinosaur? So there was only one correct answer on that one, and the correct answer is T-Rex. So let's just take a look at these here, the differences between these answers. So if you take a look, so the one in red, the first one, T-Rex, the T does not have a period after it, and that's an abbreviation, so you need the period. The second one, the one in the gold, T-Rex, that's a type of beverage, not an abbreviation. The blue one is correct. It's in italics, so that's essential. And it's abbreviated properly. And notice that the genus is capitalized and the species has a lowercase letter. That's why the one in the green is incorrect, because the species has a large R, and it should be a little lowercase r. So shift up the results a little bit. Number 3, in your lab report, species name should be. So either is okay, underlined or italicized. My preference is that when they're typed, they're italicized, and when they're handwritten, they're underlined. Okay, good work, Tristan. Number 4, which of the following ways shows the proper way to name a virus? Okay, that one was a little more difficult. Virus names always have the word virus in it, so we can exclude the Spanish flu and COVID-19. COVID-19 actually is the name of the disease, not the name of the virus. It stands for coronavirus disease 2019. Herpes virus is not a hyphen in there and not usually italicized. So that one is sort of almost okay. So the correct one is really rabies virus. So a little bit more difficult on that one. Okay, it looks like Tristan is going in hot. One more. A hypothesis. So all of the above. So good job, everyone. All right, scoreboard. Nina with the bronze, Trevor with the silver, and I think we know who's got the gold. All right, well done. Very good, very good. So I'm going to do my best to try to do one of these per lecture. I think my plan is to try to sort of have it mid-lecture, but sometimes it's going to be at the beginning to kind of, prime our memory of what we talked about last time. So just have your phone or tablet or whatever ready, and we'll do our best to have these. Sometimes I might skip it over if I'm feeling like getting a little bit behind. Okay, so back to the PowerPoint and back to a little bit about science. This is just kind of a little for your information. So I thought it was cute there, throwing in the T-Rex. The T-Rex is actually a children's book, and you can see that Tyrannosaurus is coming for tea with the little girl and the little boy. But it turns out there's actually a number of other animals that have the same abbreviated name, T-Rex. And you can see there's a whole bunch there. So we've got a rat and a shrimp and a snail. I thought that was kind of interesting. And there's a few other things called T-Rex out there, restaurant and a band and those kind of things. So just a little bit of fun facts for you. Okay, so let's talk about hypotheses. I wanted to show you this one here. So this was a doctor, I can't remember the exact year, but in the 1800s, early 1800s. And there were a lot of women dying in his hospital, and they were giving birth, and they would die from infection after birth. And so, interestingly enough, he tested many hypotheses, and you'll see that this is kind of early on in our understanding of germ theory, and you'll see some of his hypotheses are a little bit weird. But this is why, using the scientific method, he was able to test them and see which ones were false. So I'll show you some of the ideas he came up with. So one of the ideas was that the women were on their backs, so they tried on their sides. That didn't seem to improve the data. Two, there was a cathedral or a church nearby, and there was a priest that would call people to prayer and ringing the bell, and he asked the priest to very kindly, maybe, can you just not ring the bell for a couple of days? And I'm just, I'm worried about these women. That was not also true. He was worried about a poisonous gas. And that one was a little harder to test, but they can improve the ventilation. What about onset of lactation? Again, kind of a poor understanding at the time of some of the biology. Overcrowding, and then that the fever was caused by doctors' unclean hands. So how do you test this? You make them wash their hands. Now this seems like a really, really crazy idea, right? Washing your hands, but this is back in the early 1800s. People did not know there were things called germs. They did not know about that microorganisms could cause disease. So he made the doctors under his supervision wash their hands, and the number of deaths from infection plummeted dramatically. And this is great, except for the fact that he got fired after this because people didn't like washing their hands. So it's actually a very sad story in some ways. But you can see that he's testing hypotheses, and whether they're harebrained or not, this is the scientific method. So if you want to have a really good experimental design, what you're going to do is have a number of things as a part of that design. So the first thing is you're going to have an experimental group. So if you think about poor Ignace there, the group was, of course, the women who were coming in to have babies. And if you take statistics, you'll find out that having a big group is good, right? If you're just testing one subject, your data may or may not be very good. So usually the idea is the more is better. Most experiments will have at least 30 subjects or whatever, right? So, it could be mice you're testing, it could be sunflowers, all sorts of other things. Other things that you're going to do in an experiment is you're going to have variables. And variables are things that are changed or manipulated, right? So if you take a look, I have two main variables to talk about, right? First one is the independent variable. So the independent variable is often, sometimes people will call it the manipulated variable. Manipulated variable. And the dependent variable is usually what's being measured. So sometimes people call it the measured variable. Measured variable. So we're going to talk a lot about these in the lab. You know, which is the independent variable, which is the dependent variable, and so something to think about a little bit as we get to those lab exercises. So I have a couple other things to consider. Usually, when you're doing an experiment, you'll also have some sort of control. So what does that mean? It means this is the a group that you're not experimenting on. So if you were testing, let's say you're testing, you want to grow sunflowers and you want to test a fertilizer. So the controls would be some of the flowers that you're doing everything else the same. You're giving them the same amount of water and sunlight and all that, but you're just not giving them the fertilizer to see if the fertilizer has a difference. So it's kind of like your baseline. That's what a control is all about. Sometimes people will talk about having negative controls or positive controls. So the negative is where nothing is done. Sometimes you might have positive controls. So in the case of the sunflowers, maybe you have another fertilizer that you've been using for years. So you're going to test it, and you're going to test the new fertilizer in comparison to that. So you know that this fertilizer does work, but you're just hoping maybe that the new one works better. So not all experiments necessarily have positive controls, but all good experiments do have negative controls. So I'll show you another example of experimental design. Again, another historical example dealing with infectious diseases. And so this is years after Ignaz, and this is somebody who was trying to make that connection between infectious diseases and microorganisms. So his name is Robert Koch. He was a German guy. He lived about the same time as Louis Pasteur. We talked about Louis last day. And he was interested in something called anthrax. So anthrax, by the way, is a microorganism. It's a bacterium. It's found in the soil, and it forms something called endospores, which we're going to talk about. And a lot of animals can get it from the soil. It's not found all the time in the soil, but it's the kind of thing that will spread around in the soil. And animals that eat grass will sometimes inhale it up their nose, and they get it in their lungs, and it makes them sick and die. Nowadays, we have antibiotics and vaccines, and this isn't really a very big issue at all. But back 200 years ago, 150 years ago, farmers, yeah, this was a big deal for farmers, you know, having all your animals die. So he was asking the question, he was looking at these animals. You can see there's a bison, by the way. I have a bison because there are bison herds in Alberta, and some of them a few years ago actually had, there was an outbreak of anthrax. So he was looking at these dead animals and looking at their lung tissue, and he found these bacteria. And his hypothesis is that I think this bacteria causes the anthrax. So that's his hypothesis. This is explaining where the anthrax comes from. And so he's like, okay, how can I test this hypothesis? So he did some kind of relatively clever experiments, and very clever in that he had to invent a lot of methods for handling bacteria. In fact, the Petri dish, that was one of his co-workers invented the Petri dish because it had not been invented at this point. So if you take a look what he did, you can see step one here is really just what I had told you already, in that he noticed that this bacterium was associated with the sick and dead animals. So the next thing he did is he took these bacteria and he grew them up in the lab in a pure culture, and then he introduced them to new healthy animals. And the healthy animals died, and he noticed the same bacteria was found there. So this is very, very simple, but this is the kind of thing that had not been done. And like I said, you know, being able to culture these organisms in pure cultures, that kind of thing had never been done before. And so just want to kind of break this experiment apart and think about these variables here. Let's see if I can erase this and get that out of the way. There we go. Okay, so let's just answer these questions about this, right? What is Robert Koch's hypothesis? So if you think about his hypothesis, it just had to do with the bacteria. So his hypothesis was that the bacterium isolated caused illness. So in this case, anthrax is the name of the illness. And in the animals. So next question is, what was the experimental group? So the experimental group was mice. Okay, I don't know exactly how many mice he tested, but he had a whole bunch of them. And the control group were more mice. And these were mice that were not exposed to the bacteria. So maybe I'll put for the experiment group, mice exposed to the bacterium. The control group is not exposed. So you can imagine in his lab or whatever he was working, barn or whatever, he would have these mice and they were all fed the same food, given the same water. You want to have everything exactly the same except for that one variable, which is the bacterium, because of course you want to make sure you're ruling out that it's not the water or the food or the amount of sunlight or whatever else they're getting exposed to. So that being said, what are the variables? Two variables. Independent variable is the presence or absence of bacteria. So this is what the experimenter is manipulating. So presence or absence of bacteria. And what is he measuring? He's measuring the presence or absence of disease. Okay, so hopefully that was relatively straightforward. These are the kind of questions I might ask you on a test or a lab. And so, asking you what the variables were. So just keep that in mind. Okay, so a couple other things to say about science. First is, can we always use the scientific process? That's an interesting philosophical question, and I'll give you some examples here, right? Some things are really relatively easy to try to understand. Does the bacteria kill the mice? Does the drug work? Does the fertilizer work? What about sort of, these types of questions here? How did the universe form or life originate, or how did dinosaurs go extinct? Because I can't necessarily test that, can I? I don't have any dinosaurs. I don't have a time machine to see how the universe formed. But we can use the scientific method, though, to test hypotheses around these things. So you probably know the number one kind of hypothesis around the dinosaur extinction thing is that there was a giant meteor that hit the Earth and caused a lot of climate change, for example, right? So if that is your hypothesis, you can test data around this, right? So if that's your hypothesis, you can try to find a crater, a massive crater. You can look at the geological record and you can look for the types of elements that are in an asteroid and see if they are found in the Earth's geological record around the same time the dinosaurs went extinct. So it's not necessarily going to prove something, but that's not necessarily what science does. Science doesn't necessarily prove things, it supports hypotheses. It gives you an idea that something might be true, and the more science we do, the more we have an idea that we are on the right path towards truth. And that's kind of, we can use a scientific method, even if it's not quite as rigorous as something done in a lab. What about bigger ended questions? Who's smarter, Canadians or Americans, right? A lot of people have some pretty strong opinions about that one, maybe. But how do you test that? How do we test who's smarter? We have different schools. We have, different systems, even how do we even measure intelligence accurately is a big question. What about love, or does God exist? How do we test these things? Some of these things are maybe we can't find answers through science, and so that's just mainly the point I want to make, is that science is a way to get knowledge. It can't necessarily be used for everything. So last experiment I want to show you is this one here. And this is the Miller and Urey experiments on the origin of life. Kind of an interesting experiment. This was, I think, the 1970s, so 50-ish years ago. And Stanley Miller was talking to somebody about, what they thought the early Earth might be like, and thinking about the origin of life. And so they're like, well, let's, maybe the early Earth had lots of lightning and heat and all that. So they basically, hey, let's just try this in the lab. So in the lab, they, you can see up here at the top, what they did is they put some things that they thought might be in the early atmosphere, so ammonia and methane and hydrogen gas, and they threw in, they're like, okay, well, there will be lots of seawater, salt water, and so there's some heat and some electricity, and let's see if something collects. And so you can see what's happening is they're collecting the product of this, and they're sending it for chemical analysis. And they found some amino acids and a few other organic compounds. So this was kind of an exciting experiment at the time, because they thought, oh, hey, maybe this is how life originated. But this is one of these things that's kind of interesting, because, of course, here's the question: was the scientific method used? And usually we have a little bit of a discussion of this in class, and there is a hypothesis here, but I don't exactly know from the history what the exact hypothesis was. So in this case here is the hypothesis, we can make life, because he certainly didn't make life. He didn't make anything living. He just made a few organic compounds. So if he's asking those big questions, then it's a bad hypothesis. If he's asking a small hypothesis, such as, can I make organic compounds given these conditions? Then the answer is yes, it is a hypothesis. So this is just kind of why you have to be careful about what your hypothesis is. You want to be very specific to the situation. So a few things science does not do. Science is not a form of ethics. Science itself is not good or bad. We can do good things or bad things with science. Think about nuclear bombs or nuclear electricity. It's not science that makes it good or bad. It's people. Science doesn't, say things are pretty or beautiful or anything like that. Those are very subjective kind of things. Science doesn't tell us how to use that knowledge. Like I said, we can do lots of great things with it. We can do nothing with it. Science is about the natural world. Sometimes there's some overlapping things between, religion and faiths, but generally it's somewhat of a different type of area of knowledge. Okay, so I just wanted to point you to a couple things. At the end of each topic, I usually have kind of a list of concepts. So you can see here's for topic 1, kind of, a few things to go over while you review your notes. So it's talking about, basically hitting some of the key points about things that we talked about in topic 1. At the end of each topic, I also have a terminology list. So it's really, really important that when we're done a unit, you go through this list and you understand what these things are. Because, I sometimes have definitions on midterms. And it's really important to understand what these words are so that you can progress in the course. Because if you don't know what these words are, you're going to have a hard time reading the tests and understanding what the questions are being asked. So I encourage you to go to your glossary in your textbooks and look these things up if you don't know what they are, and maybe even practice writing down what you think some of these definitions are. I'm going to show you some definition examples in the next unit or two. Lastly, I have some study questions for each of these units. So these study questions, they are not exam questions. These study questions are a little bit more open-ended, but what I do is I look at them for ideas for exam questions. So you can see, very open-ended, some of these questions, right? what are the two main types of cells? What are the three domains of life? So that actually I could build a multiple choice question around that. What are some characteristics of life? Again, I'm not going to ask you those kind of questions. You almost have to write an essay for it. I don't have essay type questions on my exams. But take a look at these afterwards, and I encourage you to go through them because, like I said, this is what I look at when I start writing my exams. And so I encourage you to use these as a study tool. So that is it for topic 1. I'm going to move on now to topic 2. And okay, I'll load that up, share that with you. Okay, so topic 2 is all about macromolecules. So I'm hoping it's going to be a little bit of a chemistry review for you. I know some people took chemistry probably recently, and for some people it was probably not so recently. Mostly we're going to focus on macromolecules. So by that I mean lipids, carbohydrates, nucleic acids, and proteins. And what I'm going to do, I'm probably not going to start it today. It'll be next day. But a lot of these lectures, I build notes, and so I might start some notes today, I might start them next day. It kind of depends on how far I get into this. And so you'll need pen and paper for these notes when we go through them. And so, you know, these notes in this case I'm going to use to kind of summarize up some of the things that we're talking about in this particular unit. So like I said, a little bit of chemistry review for you, and I'll try to point out some of the things that you need to know in terms of chemistry. One thing that you do need to know is to be able to recognize these molecules. So when you see them, I would like you to recognize, what is that? Is it a lipid? Is it a carbohydrate? Is it a nucleic acid? Is it a protein? So as we go through this, that's something to take mental note of, is how we recognize the difference between carbohydrates or proteins or whatever. All right, so, oh yeah, that's just, we're going to skip that for now. I'm going to, I'm going to email out some lab instructions tomorrow. And so just take a look at that email. Somebody has a question.
Sorry, I was just looking, where's the PowerPoint for this topic on the
I haven't posted it yet. I just finished it today, so I meant to post it, but I forgot to. So I will get it up right away after class. Good question, though. I don't necessarily always get them up just before class. Sometimes I'm still working on them like 5 minutes before class, but I'll do my best here. Okay, so let's just talk about what cells are made of. You probably know a huge percentage of cell is water. Small percentage of it is things dissolved in that water, so ions and maybe other small molecules. And then a big part of our dry weight of our cell, when you take away that water, is macromolecules. So macromolecules includes proteins, nucleic acids such as RNA and DNA, so there's more RNA than DNA in our cell. There are some sugars and saccharides, and some lipids such as phospholipids and other lipids. Phospholipids, of course, is going to be the major form of lipid. Unless you're a fat cell, then you might have other lipids in there. So chemistry review. Here's the periodic table. I've kind of cut out the bottom part of the periodic table because it's not really that important to human biology. But this is showing the elements that are essential for human life. And you can see there's 25 that are on that list, and there's a few that might be important. We're still learning about that biology. But if you take a look at these, the most important elements are these 4 here. So hydrogen, carbon, nitrogen, and oxygen. So if you are taking organic chemistry, or if you take organic chemistry in the future, you are going to learn a lot about carbon, hydrogen, nitrogen, and oxygen. Because they're very important for biological molecules. And you're going to see all 4 of those in the macromolecules we're going to talk about today and next day. So, not to scare you too much, but here's just a little bit more chemistry for you. You may remember this word valence. This is showing the valence of those 4 elements. So by valence, I mean how many electrons are able to bond in the outermost electron orbital shell. So if you take a look at hydrogen, it has one electron able to bond with something else. If you look at oxygen, it has two that are free and ready to go. Nitrogen has three, and carbon has four. A little bit of chemistry for you. So you're gonna see that when we look at these molecules. You're gonna see how all of these things are going to link together. So something else that's important for chemistry and biology is this concept of functional groups. So I'm going to point out some of these functional groups when we talk about these different macromolecules as a way to help identify them. And probably the most important one we're going to see actually is this one here, the hydroxyl group, which is also called an alcohol group. So it's an OH, so an oxygen and a hydrogen. And that's found in actually most of the macromolecules, but most important in carbohydrates. There's also carbonyl groups. Carbonyl group is this carbon double bonded with oxygen. There's a carboxyl group. So this is this whole group like that. That's an acid. There's amino groups. So we find this in amino acids. Sulfhydryl, probably won't mention that one again, but it's found in some amino acids. And phosphate groups, which are found in nucleotides mostly. And we also find this in ATP. So we'll watch out for some of those. I'm not going to ask you on a test what any of these are, but I might point them out in lecture. So what do we have for macromolecules? It's kind of like the four food groups. We have lipids, so that includes things like fats. We have carbohydrates, so also known as sugars. We have proteins, and also known as peptides. And the last food group, we have nucleic acids. So that includes DNA, RNA, and also some energy nucleotides such as ATP. So before we actually get into them, I just want to introduce one other thing, a little bit of chemistry in terms of how these things generally link together and get detached. So most of these macromolecules will form things called polymers. So you can see I have a definition there for you, right? A polymer. Probably when you think of polymer, you're thinking of something like plastic. Plastic is made of basically carbon molecules like ethylene, except for there's many of them linked together. And that's what a polymer is when you have a bunch of little things linked together and they're the same or similar. So these polymers are linked together, and they're linked together through something called dehydration synthesis. So I'll show you how that works. But if you take a look here, here's our little short polymer, and we've got one monomer. A monomer means one. And what we have here is HHO. So if you count that up, that's H2O. And so removing that, that H2O, that is a dehydration. And there goes a little water molecule. And then what happens is basically the bond that was here, or the electron that was there, and the electron that is there come down and they form their own little bond and form this polymer reaction. So this is called dehydration synthesis, or sometimes people call it condensation synthesis. Our textbook is calling it dehydration, so I will try to be consistent with that kind of terminology. So what if you want to break the polymer? It's actually the exact reverse reaction. So if I want to break this bond here, then I can introduce a water molecule. And this is called hydrolysis. So hydrolysis right there, hydrolysis. So hydrolysis, hydro means water, and lysis means to break. So this is a water breaking reaction. So probably see me use that terminology, and you'll see this in a lot of the slides as these polymers are being put together and linked or whatever. And so I might get a chance to show you an example here in a moment with fats here and lipids in a moment. So as I mentioned, I'm going to try to show you what some midterm questions are going to look like as we go along here in the next couple of weeks. Sometimes I have definition type questions on my midterms, and usually the way it works is I'll grab a handful of words, maybe let's say 8 words, and you have to answer 5. So you pick your favorite 5 or the ones that you think you know the best about, and you gotta give me a definition. And so what I'm looking for in these definition type questions is that you don't give me one word, And it doesn't have to be a sentence. On my test, you don't have to use sentences. You can use point form, but a sentence is fine. But what I want is kind of like a complete thought. So you may not get a full mark if you're not giving me sort of that complete idea. So polymer, we just talked about what a polymer is. So here's an answer of what a polymer is. And you can see what I've done. I've broken it down into kind of 2 half marks, right? So you better say something about a molecule, right? So that's probably maybe a quarter of the full 1 mark, right? It has many similar or identical building blocks linked together by covalent bonds. That may not necessarily be the only definition I will accept. Always a good idea to give me examples. So maybe you didn't have this part here, but maybe you gave me an example and you say, okay, a polypeptide, that's a protein. Or maybe another example would be cellulose is a polymer. Cellulose is a polymer of glucose. I know we haven't talked about cellulose yet, but we'll get there. But what I'm saying is examples are good. Examples are always great. And not just in my class. You know, when you go through your whole education, answering exam questions, giving examples, when you write essays, giving examples, it's a great way to kind of, you know, support your argument or whatever it is you're trying to do. So I'm going to spend, I got about 3 minutes left. I'm going to spend about 3 minutes just kind of talking about lipids and taking a look at some structures there, and we'll pick this up next day. We'll come back to it. So first thing to think about lipids is, what is a lipid? So should have left a little bit more room here, but I'll write a definition for you. As a lipid is a biological molecule that is hydrophobic. So you probably, I'm hoping everyone knows what hydrophobic is. It means it does not dissolve in water. Sometimes we might use the same with the word nonpolar. And I know that's not a very specific definition, but that's kind of the problem with lipids, is that there's many different types. And so it's a little bit hard to define them any more than that. And you'll see these different types I'm going to show you. Some of the structures are very, very different from one another. This very first structure I'm showing you on this slide, these are fatty acids. And if you take a look at a fatty acid, it's just full of hydrogen and carbon, almost completely full of hydrogen and carbon. So you may remember, again, some chemistry, that carbon and hydrogen, their electronegativity is almost identical. And so that is a nonpolar molecule. And nonpolar molecules are generally hydrophobic, meaning they don't mix with water very well. Now, there are a couple of little oxygens here on the end. They're not enough to make that fatty acid polar overall. And in fact, that little group at the top, that's an acid group. It's a carboxylic acid. So that's where it gets the name. So if you take a look, there's my label. You can see the carboxylic group is at the top, and this big chain at the bottom, we often call it a hydrocarbon chain, and it's very, very nonpolar and doesn't mix with water very well. Notice the one on the right is a little bit different. It has some double bonds in there. And these double bonds, sorry, I'll just erase that out of the way so we can see what that says. These double bonds make this thing unsaturated. So what does that mean? The one on the left is fully saturated, meaning it has lots of hydrogens. The one on the right, you can see if you take a look, this is not a CH2 anymore. It's a CH, so it's missing some hydrogens because of the double bond. So it's kind of the terminology that they came up with. I don't know why. I would have probably named it something else myself. But the big difference between saturated and unsaturated, I'll show you on the next slide. You can see I have this note about them packing close together. So saturated fatty acids can pack really well together. And so what does this mean? It means that there's lots of London dispersion forces, and they can be more solid at room temperature. So usually when we talk about saturated fats, we're talking about things like butter or margarine, and the word we use, especially if we're talking about things in our diet, the word we use is fats. Things that are unsaturated, I'll just go back to that previous slide for a second. You can see that the the double bonds kind of make it kinky in that it's kind of, there's lots of kinks in there. And so when these things are together, they don't really pack very well. And so there aren't very good intermolecular interactions. And so often these end up being liquid at room temperature. And when we're talking about food, we call them oils. So let me just take a peek here. So that's actually a good place to stop for today. Sorry I went over just a minute there, but we'll come back to lipids. I'll redefine them next day. And like I said, be prepared to take some notes. I'm going to start writing some notes for you next week. So make sure you have a pen and paper. So hopefully everyone has a wonderful Wednesday, and I will see you on Friday.